Hi, everyone. Today, we're going to continue on a relatively shorter chapter, chapter 10, and we're going to talk about enzymes. First, enzymes are proteins. So that's why we study enzymes right after we study the protein chapter. The reason we pick out enzymes as a separate chapter to study because enzymes are so important in helping biological reactions to proceed or to be performed in cells under very mild conditions, such as near neutral pH, such as near body temperature. Okay, you don't need high temperature, you don't need heat or acid or base. These reactions, the biological reactions inside our cells, are actually carried out under very mild physiological conditions. And those reactions are helped by the enzymes. So that's why we have tons of reactions in our cells. So that's why we have a lot of enzymes that are responsible for the function. So what are enzymes? Well, enzymes are proteins that act as biological catalysts, or we call them biocatalysts. What are catalysts? Catalysts basically are entities, chemical entities that accelerate chemical reactions. Okay, accelerate chemical reactions. So basically making our biological reactions to proceed in a very smooth and fast and efficient way. And meanwhile, these reactions are carried out under very mild conditions. So that's the function of the catalyst, to accelerate chemical reactions. And for biological catalysts, enzymes, they accelerate what? Biochemistry reactions. And some background, the name enzyme is derived from a Greek word meaning in yeast. It indicates that these catalysts are present inside cells. And also, almost all metabolic processes in the cell needs enzyme catalysis in order to occur, at least in order to occur at rates fast enough to sustain life. Okay, that's why we said we have to learn these types of biomolecules so that help all these reactions we described so far, no matter it's protein synthesis, carbohydrate synthesis, carbohydrate hydrolysis, or changing from one carbohydrate to another, or sometimes helping protein to fold, etc. They all need enzymes, okay, for the reactions to happen. Now, how does enzyme work? Or this picture actually show you how does a catalyst work. Enzymes are biocatalysts. Enzymes, just like any other chemical catalyst, the same, they speed up or accelerate chemical reactions by lowering the activation energy. What is activation energy? This chart shows you that. If a reaction needs to happen, the reaction has to go over a barrier. We call that energy barrier. And that energy barrier, before you go over that energy, after you go over that energy barrier, the reactants can be converted to a product. And that barrier, like a hill, the reactants must go over the rise of the energy to go over that energy. That barrier between the reactants and the highest level of that barrier is called the activation energy. So it needs to overcome that energy to become products. That's for a reaction to happen. All reactions are like this. But with enzyme or with a catalyst, the barrier is now lowered. Okay, there's a lot of mechanism involved. Okay, most times enzyme coordinate or participate in that reaction. So the barrier is lower. So you can see that with enzyme, okay, with enzyme or with catalyst, the barrier is lower, okay, lower than without. the enzyme. If the barrier is nowhere, means what? Means it's much easier for a reactant or for the reactants to overcome the barrier to become products. And because of that, the reactions are faster. So what happens is enzymes lower the activation energy, lower the energy barrier for the reactants to overcome so that the reactions are faster than without enzyme catalysis. Okay, this is how, in general, how enzymes work. And enzymes are like proteins, can be classified into two types. One type of enzyme is simple enzyme, means just protein, no other parts, just amino acid peptide chain. But a lot of enzymes are conjugated enzymes, meaning the enzymes are combining with other organic or inorganic components, just like conjugated protein we studied in chapter 9. And those enzymes are called conjugated enzymes. Now, in conjugated enzyme, the protein part, the peptide chain part, is called the apoenzyme. Okay, the apoenzyme is just the protein peptide chain part. And the other components, the non-protein part, is called the cofactor. Okay, it's called a cofactor. So a conjugate enzyme is basically a apoenzyme conjugated with what? With a cofactor. With a cofactor. And like I said earlier, cofactor, just like the prothetic groups in chapter 9, we study prothetic groups for conjugated proteins, can also be organic or inorganic. Okay, organic or inorganic. If the cofactor is organic, if the cofactor, the non-protein part for the enzyme, is organic, we call the cofactor a coenzyme. A coenzyme. Okay, organic molecules, cofactors are called coenzymes. A lot of vitamins, except vitamin C, have functions as coenzymes in the human body. Now, of course, if the cofactor is inorganic, we call them cofactors. And most inorganic cofactors include the metal ions, such as zinc ion, magnesium ion, manganese ion, and iron 2+ ion. These are inorganic metal ion cofactors. So these are the composition of an enzyme. Okay, most enzyme contains the protein part called apoenzyme and conjugated with something called cofactor. And cofactor can be inorganic or organic. If it is an inorganic molecule, we call them cofactors, mostly metal ions. But if it is an organic molecule, we call that cofactor a coenzyme. Mostly we call them coenzymes. And coenzymes mostly are vitamins, especially vitamin B. Okay, especially vitamin B. So that's the composition of enzymes. Next, let's take a look naming enzymes and also how do we classify enzymes, because we have so many enzymes in our biological systems. First, enzymes. Okay, when we name an enzyme or when you look at the names of these biological entities, enzymes end with ase, ase. Okay, so the suffix ase identifies a substrate as an enzyme, something as an enzyme, ase. And enzymes are categorized by the type of reaction the enzyme catalyzes. So the type of reaction will serve as a prefix for the ase, okay, for the suffix ase. For example, oxidation reaction, if it catalyzes oxidation reaction, we call that oxidase. If it catalyzes hydrolysis reaction, we call them hydrolase, etc. So the type of reaction will classify different enzymes, and that type of reaction will serve as a prefix for the ase, for the enzyme. And also, sometimes we need to note the substrate or the reactant the enzyme acts on. Remember, enzyme catalyze what? Reactions from reactants to products. So the reactants, the enzyme act on, is also noted in the name. So for example, glucose oxidase means what? An enzyme oxidizes glucose because the enzyme acts on glucose. So glucose is the substrate of the enzyme. So the identity of a substrate is often noted in addition to the type of reaction. So glucose oxidase means what? The enzyme oxidizes glucose. The reaction will be glucose will be oxidized, and the enzyme is glucose oxidase. So this is how normally we name an enzyme. Okay, normally how I say these are the parts for naming an enzyme. Now, most importantly for the naming part, I need you to know the main classes of enzymes. Okay, the main classes of enzymes and the type of reaction each class of enzyme catalyzes. Okay, this table shows you the main 6 classes of enzymes. Okay, there are, of course, each class there are subclasses, but you can understand them as different types of reaction that catalyzes each class. But most importantly, know these 6 types of or 6 classes of enzymes and the type of reaction each class catalyzes. Okay, it's in the red box, shows you the type of reaction each classes catalyzes. So I'm going to go over these 6 classes. I'm going to give you examples of each class of enzyme. What type of reaction do they catalyze? Again, the type of reaction catalyzed is listed in this red box. You can always refer to, hey, what does the reaction look like? If the reaction falls into this type, what type of enzyme we're talking about? Okay, so these 6 main classes are oxidoreductases, means catalyzing what? Oxidation and reduction reaction. The second type, transferases, means the reaction transfers something. The enzyme will be responsible for transfer something. Number 3, hydrolases. Okay, hydrolases means what? The reaction is the enzyme catalyzes hydrolysis reaction. Lyase is a unique type of enzyme that will catalyze, remove some small molecules from a substrate, such as remove water, remove carbon dioxide, etc. Remove some part from a molecule. Number 5, isomerase. Okay, isomerase, if you remember that word isomer, when we study in the first 2 chapters, isomer means what? 2 molecules with the same molecular formula but different structure. So isomerase basically creates isomers. A becomes B, but A and B are isomers. And last one, ligase. Ligase, this word came from a word ligation. Ligation means joining. Okay, ligation means joining. So ligase basically means function of joining substrates, putting 2 together. Okay, making a new bond between 2 substrates. Okay, 2 substrates. Okay, so these are the 6 main classes of enzyme. I need you to know their names and these classes. And again, you can always refer to the type of reaction catalyzed for each main class. Okay, with that, let's take a look at reactions for each class. So you kind of idea what the enzymes are doing. The first type, like we mentioned, is oxidoreductase. Okay, oxidoreductase. This type of enzyme basically catalyzes what? Oxidation and reduction reaction. We said oxidation and reduction reaction. Oxidation and reduction. Okay, reduction reactions. So what is oxidation and reduction? If you remember, we learned before, oxidation reduction basically means what? Adding hydrogen or removing hydrogen. Removing hydrogen is oxidation, adding hydrogen is reduction. So basically what oxidoreductase does is it will help the introduction of double bond by formal remove of 2 hydrogen atoms from a substrate. Okay, remove hydrogen to make a double bond. If that's the case, the reaction is catalyzed by an oxidoreductase. Of course, the hydrogen being removed will be accepted by a coenzyme, okay, such as NAD+. Okay, NAD+. We're gonna study what is NAD+ in chapter 12. It's a coenzyme, okay, helping getting the hydrogens removed. That's why NAD+, you can see that becomes NADH. Okay, it gets a hydrogen that is removed from the substrate. But take a look at the substrate. The substrate is alcohol. 2 hydrogens are removed from the C and the O. That's why you got a CO double bond. Okay, a hydrogen removed from the C, a hydrogen removed from the O, you get a CO double bond. That is the definition of oxidative reductase, by removing 2 hydrogens from a substrate and making a double bond. And hydrogen, again, is accepted by a coenzyme. That coenzyme here is NADH. Okay, NAD+, and then becomes NADH. So this is a reaction from oxidative reductase. Okay, again, guys, always refer to that. You can see that introduction of a formal double bond by removing 2 hydrogen atoms and introduction of double bond and by removing 2 hydrogen atoms, and hydrogen atom is accepted by a coenzyme. So always refer to the table, okay, later on. Okay, the second type is a transferase. Okay, transferase. Transferases transfer a functional group to another substrate. The most commonly seen transferase here is, you can see that this transferase transfer a phosphate group between 2 molecules, between 2 substrates. Okay, phosphate group. You can see sugar has an OH, and the enzyme transfer a phosphate to the OH, becomes a sugar phosphate. And where does the phosphate come from? From this molecule called ATP, adenosine triphosphate. So the enzyme transferred 1 phosphate from ATP to the OH of the sugar. When ATP transfer a phosphate, ATP, the triphosphate, will become a diphosphate. That's why it becomes ADP. Tri-di, missing 1 phosphate. Where does phosphate go? It went to what? To the sugar. You can see that. So basically what happens is the enzyme transfer 1 of the phosphate from ATP to sugar, and ATP becomes ADP, adenosine diphosphate. Okay, that's a transferase. Okay, again, we're going to study ATP and ATP in chapter 12 as well. They're very important metabolism intermediates. Okay, but the enzyme is a transferase, transfer a phosphate now from ATP to sugar. Okay, again, that's an example. There are other transferases. I need you to identify the reaction, be able to identify the reaction. What type of enzyme is catalyzed? Number 3, hydrolases. Hydrolases basically means catalyze hydrolysis reactions. Okay, hydrolysis reactions. For example, in here, hydrolase, this hydrolase catalyzes the hydrolysis of a disaccharide into 2 monosaccharide. You can see that. This is a disaccharide, and now you get 2 monosaccharide. That means what? The bond, the glycosidic bond, is cleaved. Okay, cleaved, and water is added, by the way. Okay, hydrolysis, you need water. So water is added to the 2 molecules. That's why it's hydrolysis. Okay, and that enzyme is a hydrolase. And by the way, a lot of digestion enzymes are hydrolysis enzymes, hydrolases. Why? Because digestion, think about it, is digesting what? Carbohydrates, digesting what? Proteins, digesting what? Digesting lipids. All these molecules, if they want to break down into small molecules for us to absorb, they need to be hydrolyzed. So a lot of digestion enzymes, hydrolyzed proteins, hydrolyzed lipids, hydrolyzed carbohydrates here are hydrolases. Okay, digestion enzyme hydrolases. Next, lyase. Okay, lyase. Remember I said earlier, lyase is you need enzymes basically remove a small molecule or sometimes can add a small molecule to a substance. Here, it's a very common reaction for lyase is removal of water from a substance or addition of water to a substance. Okay, here you can see that add a water to the double bond, you get OH and H. Or you can call it remove the OH and OH and OH to give a double bond. That's the same reverse reaction. They're both catalyzed by the same type of enzyme called lyase. Okay, remove OH and H to make water, or add water to a hot double bond to give a H and OH. And that reaction is catalyzed by a lyase. Okay, lyase. Of course, you can see if you look in the lyase, you can see that there's other reactions lyase can do. Remove of water, remove of CO2, remove of ammonia, and also addition of water to a substrate. They're all catalyzed by lyase. Again, always refer to the table to check what type of reaction each type of enzyme catalyzes, and then you can recognize that reaction. Next, isomerase. Okay, again, isomerase by the prefix isomer means what? The enzyme creates isomers. What are isomers? The same formula, but different structure. So what isomerase is basically transfer a functional group, for example, in this case, a phosphate from one position to another. Only you transfer the position, you don't change what? The components, you don't change the formula. You only change the location of a functional group. If that's the case, you're getting isomers. Okay, you're getting isomers. For example, you can see here the phosphate on the left molecule is on number 3 carbon. The phosphate on the number 2 on the right molecule is in number 2 carbon. So that means what? The enzyme transferred the phosphate from 3 to 2. But everything else is the same. You can see it still has the OH, still has the phosphate, still has every other components. The molecule still has the same molecular formulas. It means how many carbon, how many hydrogens, how many phosphorus. Nothing is changed. Only is what? The position of the phosphate is changed. So these two are called isomers. If the enzyme changes to isomer, then what? Then the enzyme is an isomerase. Okay, isomerase. Another way I can show you is how do you recognize the type of reaction? Enzyme just change from AB to BA. It's the same thing. You change the position of something, then that's isomerase. And finally, ligase is from the word ligation, means joining two species. In this case, you can see that the enzyme catalyzes the joining of this carbon and the carbon dioxide carbon. So you basically make a new CC bond here. A new CC bond here, joining the C and another C to make a new CC bond. And this reaction normally will be needing energy. So that's why this a ligase reaction will need the participation of ATP. Okay, ATP. ATP is an energy substance in our cells. We're going to study that in chapter 12. ATP provides the energy. Okay, ATP in this case doesn't provide the phosphate. You can see that ATP just hydrolyzes, become of ADP and phosphate. It doesn't provide the phosphate for the reaction. It just provides the energy. Okay, energy. Provide the energy for the reaction. Okay, that's ligase, joining two species together. Okay, so these again are the 6 main classes of enzymes. I need you to know these 6 main classes and be able to recognize a reaction and identify what type of enzyme this reaction is catalyzed by using the table listed here. Okay, it's very important in this chapter. Okay, here a quick kind of like a concept check. You can see, guys, this reaction shows you why apple or some vegetable turns brown or discoloration. Okay, apple, you cut a piece, it turns brown. What happened here is the phenol derivatives is not exactly phenol, but there are phenol derivatives in apple, in some of the fruits. They react with oxygen to become quinol derivatives. Okay, quinol derivatives. So you can see that this reaction, basically what happened is you add some oxygen to the structure and also remove some hydrogen. You can see OH now becomes what? CO double bond, CO double bond. So hydrogen is removed, so that's why you can get CO double bond. If hydrogen is removed, we know the reaction is catalyzed by what? Oxidoreductase. This reaction is oxidation reaction. Okay, oxidation reaction. So basically why the apple turns brown color is because the phenol derivatives gets oxidized in the air. That's why you have to cut pieces exposed to the air. Air will oxidize these phenol derivatives. Next, let's take a look at some facts about enzymes and also discuss how does enzyme work? How does enzyme catalyze a chemical reaction? Okay, enzymes exhibit different levels of selectivity, or we call specificity for substrates, for the reaction they catalyze for. There's some kind of picking. Okay, we call that selectivity or specificity. And there are several types or levels of specificity. One level is called absolute specificity. This means one enzyme will only catalyze one reaction. Very restrictive. Okay, that's called absolute specificity. One enzyme does one reaction, not the other. That means on one enzyme only accommodate one reactants for that reaction, nothing else. That's called absolute specificity. Another level of specificity is called relative specificity. Okay, relative specificity. Relative means it's a little more relaxed. The enzyme can act on a few substrates that are structurally related. Okay, not structured. I mean, it's a few substrates, they're looking like each other. The enzyme can maybe accommodate all of them or some of them, not just one-to-one. And then that's the case. If that's the case, we call relative specificity. It's very common in our biological systems. One enzyme can accommodate a few substrates, but their structure must be related. They look like each other at least. Another level is something we have actually talked about earlier, called stereochemical specificity. Stereochemical specificity means the enzyme can distinguish stereoisomers. For example, D sugar versus L sugar, or D amino acid versus L amino acid. Remember, I said our biological system can only process D sugar. That means enzymes cannot accommodate the mirror image, the L sugars. The enzyme has an environment that is chiral. Okay, enzyme, the active site also is chiral, so that it can distinguish one mirror image versus not the other. So that's called a stereochemical specificity. So these are the level of specificity. And with that type of specificity, we want to explain why. Okay, stereochemical specificity can be easily explained because we know enzymes are made by amino acids that are chiral molecules. 19 out of 20 amino acids are chiral molecules. So enzyme has a chiral environment, and that chiral environment can recognize chiral molecules. That's very easily understood. But how can we explain absolute versus relative specificity? Why some enzyme only can catalyze one reaction? Why some enzyme can accommodate a few substrates? Okay, in order to do that, let's take a look at the action of enzyme and also the mechanism of enzymes. Okay, we're doing this in a very brief way. Of course, in advanced biochemistry class, we're going to study very details of the kinetics of enzymes, etc. But here, we want to talk about what does the enzyme look like and how enzyme accommodate substrate, how enzyme catalyze reactions. All enzymes have a place, a small part of the enzyme where the substrate binds to the enzyme and where the reaction occurs. And that place, tiny place, is called the active site. The active site. So the active site is the enzyme, is an enzyme structure where a substrate binds to the enzyme and also where the reaction takes place. So first, during the enzyme catalysis, what happened is an enzyme binds to the substrate at the active site. You can see if this is the substrate, this is the active, they bind to each other. And then they will form an enzyme substrate complex called ES. Sometimes they call ES, enzyme substrate complex. And at that active site, the complex at the active site, the enzyme will help, some amino acid side chains will help the reaction to proceed. So the enzyme substrate complex will be turned into enzyme EP, product complex, or products complex. Okay, products complex. And you can see that complex, the substrates and the products are still bound to the enzyme active site. But at the active site, catalysis occurs. Okay, catalysis occurs. And again, the amino acid side chains or some functional groups will help the reaction, will facilitate the reaction to lower the activation energy, to convert enzyme substrate complex to enzyme products complex, EP complex. And then you got the products is going to be released from the enzyme. They repeat from enzyme. And now the enzyme active site is what? Is empty again. So the enzyme active site can accommodate another substrate and go on in a cycle. And this reaction, this turnover is very fast. That is why we don't need a lot of enzymes for chemical reaction, because the enzyme is kind of like recyclable, right? After the products are released, the enzyme active site can accommodate new substrate now. So a small amount of enzyme can catalyze a lot of reactions. Okay, keep that in mind. So this picture shows you what happens in an enzyme catalysis in a very brief, fundamental way. Okay, fundamental way. Here, this picture shows you what an active site looks like and how a substrate can bind to the active site. Here, the picture shows you in a cartoon way. The forces that draw the substrate into the active sites are these three types of forces: electrostatic interaction, hydrogen bonding, and hydrophobic interactions. If you still remember what we studied chapter 9, when we studied tertiary structure of proteins, you will recall these are the types of reactions that are responsible for protein tertiary structures. So that means the substrates has some functional groups that can interact with the side chain of the amino acid at the active site. So the active site has some amino acid side chains that can interact in a nice way with some functional groups on the substrate. And those interactions are those three types: electrostatic interactions, hydrogen bonding, and hydrophobic interactions. And sometimes cofactors can also help binding as well. Cofactor will involve into that binding together as well. Okay, so in that picture, you can see that the yellow groups, the yellow groups means the interaction that binds the substrate to the enzyme active site. Okay, the green groups means the tertiary structure of the protein itself. But the yellow ones means the active site amino acid side chains, the R groups, are interacting with the substrate through these types of interactions. And also sometimes cofactors get in there too. Okay, for example, this one shows you the real active site with the substrate. Here is the substrate. Let me use a different color pen. Which one is brighter here? This is the substrate, a carbohydrate, a monosaccharide actually. And this is the cofactor, manganese ion. And these are the amino acid side chains at the active site. You can say that these are the important active site amino acid. They're interacting with the metal ion, and the metal ion interact with the substrate. So this is how a substrate is kind of like a docked into the active site with the amino acid side chains in the active at the active site, and also with the cofactor. Okay, this enzyme is called D-piscose 3 epimerase, and the substrate is fructose, and also showing the coordination of metal ion and also the substrate binding site. The purple ball indicate the manganese ion coordinating key amino acid residues. Okay, key amino acid residues. So this is a real real active site showing the protein inside the protein, where the substrate binds. Okay, again, this is fructose. Sorry. Okay, this is fructose. Fructose, okay, which is the substrate. Okay, substrate, and that's the cofactor. Okay, cofactor. Okay, the metabolite is the cofactor. So that's how enzyme works. Now, when enzyme and substrate binds, there are two theories, okay, two theories that can help explain those two levels of specificity. One theory is called the lock and key theory. Okay, lock and key theory. It states that the active site and the enzyme in the enzyme has a fixed, rigid conformation that the substrate has a complementary shape that exactly fits such a site. So basically lock and key, right? The active site here fits the substrate in a rigid way, perfect fit, rigid, fixed way. If that's the case, the enzyme can only accommodate that substrate because the lock and key, the conformation is very fixed, rigid. So that means the enzyme cannot get anybody else. And this theory explains enzyme what? Absolute specificity. The absolute specificity. The absolute specificity. Okay, absolute specificity. Another theory, which is more common actually, is many enzymes, their active site has some flexibility, which will change, okay, change to accommodate a few substrate, structurally related substrate. So basically, when the substrate approaches, the conformation of enzyme will kind of like a change to adapt themselves to accommodate the substrate. Okay, accommodate. So kind of like substrate induced the change of the substrate of the active site to accommodate themselves. And that theory is called induced fit theory. Okay, induced fit. The conformation of the active site change and adapts to accommodate an incoming substrate. Okay, that's why some enzyme can accommodate a few structurally related substrate, even though they're not the same. But the enzyme active site is flexible, can accommodate. And this is a more thorough explanation because it not only includes the specificity we talked earlier, but also talk about the flexibility of enzyme protein. Now, why enzyme has relaxed the flexibility, has relative specificity. So these are the two theories, okay, two theories. And also we explained what the mechanisms are, enzyme catalysis are. Okay, next part. Okay, next part. We're going to study what are the factors affecting the activity of enzymes. Why some factors increase the activity, some factors decreases the activity? There are four major factors. First one is temperature. Okay, of course, temperature. Enzyme functions at a certain temperature. If our body, if enzyme in our human body, the best temperature is what? It's 37 degrees, our body temperature. So each enzyme has a temperature at which the enzyme has the highest, the maximum activity. And that temperature is called the optimal temperature. Okay, optimal temperature. Optimal temperature means at that temperature, enzyme has the highest activity. And also it means when the temperature is lower or higher than the optimum activity, the activity is going to decrease. Okay, decrease. And if the temperature is too high, okay, too high, the enzyme is going to be denatured. Remember, heat will denature protein. That means high temperature will denature an enzyme. And because of that destroying effect of temperature on bacterial enzymes, hospital and labs actually use high temperature and high pressure to sterilize medical instruments and laundry, because high temperature will deactivate bacterial enzymes to kill bacteria. So high temperature enzyme can be deactivated. Low temperature, on the other hand, the activity is going to decrease, but lower temperature normally will not deactivate an enzyme. As a fact, we actually sometimes in the lab, we store enzyme in very low temperature, like -20. So that's the difference. Okay, high temperature will cause the enzyme to be destroyed, denatured. But lower temperature, normally they don't, even though the activity will be low at lower temperature than the optimum temperature. Okay, that's temperature. The second factor is similar to temperature, is called pH. Okay, pH. pH means the acidity or basicity of a system. And enzyme has its own optimum pH at which enzyme functions at the maximum rate. Anywhere lower or higher than the optimum temperature, the activity is going to decrease. And extremely low and extremely high pH enzyme both will be denatured. Okay, denatured. Remember we said chapter 9 as well. Acid and bases can potentially denature a protein. So extremely acidic or extremely basic, both cases, enzyme can be denatured. Okay, click markings here, denature. Okay, but activity is going to decrease if we move away from the maximum optimum pH. Each enzyme has a characteristic optimum pH, usually is around 7. But some digestive enzymes, for example, enzyme in your stomach, they function at very low pH, like pH around 2, because our stomach fluid pH is around 1 and 2. Okay, so this picture shows you the optimum pH of these enzymes and where they're located, the source of the enzyme. Okay, for example, pepsin is an enzyme that hydrolyzes peptide. You can see that its source is located in gastric mucosa. So the optimum pH is 1.5. Okay, alkaline 5. And this enzyme, alkaline phosphatase, the location is bone, and the optimum pH is 9.5. It's very basic. So it depends on the location of the enzyme, and the enzyme may have different optimum pHs. Okay, the third factor is substrate concentration. Okay, substrate concentration. When the concentration of enzyme is certain, so you have a certain number of calories, the enzyme, then if you increase the substrate concentration, the enzyme activity is going to increase because you have more substrate to go on for a reaction. So the activity is increasing. But that increase is not unlimited. That increasing will stop at a certain point. You can see that the increased line is going to flat out reach a plateau. Why is that? Because remember, the condition here is the concentration of enzyme is constant. That means you have a certain number of enzyme available. So once all the enzymes have bound, that means all the enzymes are occupied, then the substrate increase will have no effect on the rate. Why? Because there's no more reaction to go on, because all the enzymes are occupied now. Okay, all of them are occupied now. And we call that, the maximum reaction rate when they reach the plateau. Maximum rate means what? Means at that point, that given amount of enzyme is occupied, is all full, until there's more spot release, more active site released. The enzyme, notice the reaction would not go any higher. So at maximum rate, all the available enzymes will be saturated. Okay, will be saturated. And enzyme now work at the maximum rate. So the chart shows you the activity versus substrate concentration is kind of like a curve chart. The increase is fast in the beginning, and the increase will slow down until what? Until flat out. So the rate is going to be the maximum rate. So this is what happens when you increase the substrate concentration, when the enzyme concentration is kept constant. Okay, keep that, this is very important. Okay, the enzyme is kept constant. Enzyme cannot change. So increase the substrate, the activity is going to increase, and beginning is fast and then slow down. Eventually it's going to flat out. Okay, flat out. An enzyme, the maximum number of substrate molecules per minute by one enzyme for the reactions, the maximum number of substrate can be transformed from reactant to product per minute by one enzyme molecule under the best temperature and the best pH and best saturation, everything. It's called the turnover number. So you can see that enzyme, how much substrate, how many substrate can actually saturate the enzyme. And that saturation is called the turnover number. Turnover number defined by what? One enzyme, 1 minute. How many substrate it can turn over, can change from reactant to product. You can see different enzyme has different turnover numbers. You can see, for example, in an extreme case, this carbonic anhydrase makes an enzyme making carbonic acid, combining carbon dioxide and water, can turn over 36 million carbon dioxide and water molecules per minute. Can you imagine the efficiency? This catalase that break down hydrogen peroxide, our blood has this catalase. Okay, if you drop one drop of your blood into a hydrogen peroxide solution, you will see a lot of bubble come out, because the catalase the breakdown of hydrogen peroxide solution you can purchase from a grocery store. And that's fast too. You can see that per 1 minute, one enzyme can turn over 5,600,000 hydrogen peroxide molecules. So very, very fast. Of course, some enzymes, they turn over, right? It's not that big. For example, DNA polymerase synthesize large DNA molecules, only turn over 900 molecules per minute. But DNA synthesis is a much more complicated process than break down small molecule like hydrogen peroxide. So that's called turnover. So that means how many molecules can saturate the enzyme? Okay. The last factor, okay, not really called a factor. The last factor is if you just don't limit the enzyme concentration, you increase the enzyme concentration. So that means you give as much as enzyme you want, then the activity can increase as much as you want. The activity of enzyme increases as the concentration of enzyme increases. There's no doubt, because you have more enzymes. Okay, you have more binding site, and you can bind as many possible as substrates you want. Okay, more available enzyme is available to bind the substrate. So the reaction can increase as much as you want. Depends on how many enzymes you supply. Okay, so those are the factors that affect enzyme activity. And then In the last part, we want to study another factor, not really factor again, that can cause the enzyme activity to be decreased. Okay, to be decreased. And especially we add some chemical substances that will have this effect, decrease enzyme activity. There are some substances that can do that. And we call that process enzyme inhibition, decrease inhibit, right? Inhibition. And the little substances, the small molecules or big molecules that cause enzyme activity to decrease are called inhibitors. Okay, called inhibitors. Enzyme inhibitors. Enzyme inhibitors basically decrease enzyme activity or sometimes permanently stop the enzyme activity. Now, what an inhibitor does is bind to an enzyme and cause enzyme to have lower activity or stopped enzyme activity. Depends on how the inhibitor binds to the enzyme, either permanent binding or temporary binding. We classify inhibitors into two big classes. One class is called irreversible inhibitor. That means these inhibitors bind to the enzyme almost permanently, tightly. Okay, you can say bind tightly, means permanently. Okay, permanently. And you may ask, how does an inhibitor binds to an enzyme permanently and irreversible inhibited enzyme? Through covalent bonding. Okay, through covalent bonding. So irreversible inhibitors basically inactivate enzyme, stop enzyme activity by forming a strong, tight covalent bond to an amino acid side chain group at the enzyme active site. The inhibitor covalently, permanently binds to the side chain at the active site and one of the side chain at the active site. So enzyme, the side chain is blocked. So an enzyme cannot accommodate the substrate, etc. anymore. So enzyme is permanently deactivated. Why permanent? Irreversible. Because covalent bond is very strong, it's very tight, it's not easily broken. So that inhibition, that binding is permanent. Okay, binding is permanent. An example shown here, shown here is penicillin, antibiotics. Okay, they kill bacteria by what? By binding, covalently binding the serine residue. Okay, serine residue of a bacteria enzyme called transpeptidase. Okay, this enzyme is important for bacteria to synthesize their bacterial cell wall. But penicillin binds to that enzyme at the active site, and in particular, that beta-lactam brain of penicillin antibiotics binds to the serine OH residues to form a covalent bond. You can see this is the covalent bond. This is the serine, and that's covalent bond. And because of that, penicillins are irreversible inhibitors for the bacterial transpeptidase. Okay, that's what happens. Another example shown here is the nerve gas DIFP, diisopropyl fluorophosphate, okay, which is a nerve gas. Okay, nerve gas. This nerve gas, the phosphate, binds to the phosphorus, the phosphate, binds irreversibly or covalently to the OH group of certain enzymes as well. Okay, certain enzymes as well. You can see that this is the active site serine OH group. It will make a covalent bond with the phosphate of the nerve gas. So these are toxic gas because they permanently shut down enzyme. And such gas may include choline esterase. We talked about earlier about choline. Okay, if you guys remember choline, acetylcholine, about the nerve and muscle junction. If you remember, if you can pause the video and go to chapter 6 to study that. Okay, nerve gas actually targets choline esterase. Okay, choline esterase. So these are two examples of permanent or irreversible inhibitor. They bind to the active site through what? Covalent bonding. Tight covalent bonding. Another type of inhibitor called reversible inhibitors. They don't bind covalent bond. They're not covalent bonded with the active site. So they can dissociate from the enzyme. So their binding is temporary, right? They can get away from the binding. So those inhibition is irreversible inhibition, not covalent bonding. Now, depends on the location of the reversible inhibitor, where it binds to the enzyme. We further classify reversible inhibitors into two types, depends on the location it binds. Some reversible inhibitors binds to the active site. Okay, some binds to the active site, other sites other than the active site. The ones that binds to the active site are called competitive inhibitors. The ones that don't bind to the active site, bind to somewhere else, are called non-competitive inhibitors. And later on, we're going to study non-competitive inhibitors actually are involved in enzyme regulations. But again, here, reversible inhibitors means these inhibitors are not making covalent bond with the enzyme. They just temporarily bind to the enzyme. So that's why they're reversible. But some reversible inhibitor binds to the active site, and we call those competitive. Okay, you will very easily understand why it's competitive later. And some don't bind to the active site, but some other places, we call them non-competitive inhibitors. Okay, let's take a look. Competitive inhibitors. Why they're competitive? Because they bind to the active site. When they bind to the active, what does it mean? It means the substrate cannot bind anymore. This is the inhibitor. And the substrate has to be kicked away because the substrate cannot bind to the active site anymore. So they're competing. Competing with what? The inhibitor is competing with the substrate. That's why we call them competitive inhibitors. Now, the reason competitive inhibitors can bind to the active site, you can actually guess. The competitive inhibitors would need to what? Closely resemble the structure of the substrate. They need to look close together. They need to look alike. Otherwise, how can they compete the same spot? So competitive inhibitor basically looks alike substrate, resemble the substrate. So they compete with the substrate at the binding site. They prevent the substrate from approaching the binding site. That's why you have inhibition. And we know the binding is not permanent, it's temporary because it's reversible. So if you increase the substrate, you got more competitors, you get more substrate, then the substrate can actually kick it out. Okay, increase the substrate, then you can reverse the inhibition. Okay, reverse the inhibition. That's how it works. Again, the inhibitor resemble the substrate. The inhibitor binds to the active site, so the substrate cannot bind. If you want to reverse it, you increase the substrate concentration. So get more thing to kick the inhibitor out. Now, competitive inhibitor is actually very important because competitive inhibitor resembles substrate. So it gives what? Gives us some information of the active site. Gives us information how does enzyme active and the substrate bind to each other. How do they interact at the active site? Because remember, the inhibitor resembles the substrate. So it provides a lot of structural information regarding the binding, the initial binding. And because of that, a lot of pharmaceutical companies have synthesized drugs that competitively inhibit metabolic process such as bacteria or some cancer cells. Why? Because from the inhibitor, we know how, kind of have a clue how the substrate binds to the active site. Then we can have some structural information to design some better inhibitors to the enzyme of bacteria or enzyme of cancer cells. And these drugs can serve as the inhibitor of these cells or bacteria. Okay, one example, very classic example, is sulfur drugs. Okay, sulfur drugs. Many bacteria needs this guy, p-aminobenzoic acid. Para means 4-aminobenzoic acid, to produce folic acid. Okay, to produce folic acid, this bacteria needs that. But a class of antibiotics called sulfur drugs, the sulfonamides, it resembles this p-aminobenzoic acid. You can see these two structures. They look like very much, right? They're very much close to each other. So sulfonamide compete with this PABA, P-A-B-A, this p-aminobenzoic acid, competed at the enzyme active site. So to prevent what? Prevent enzyme bacterial growth, because bacteria cannot synthesize their own folic acid. And sulfur drugs only affect bacteria because we can get folic acid from our diet. We don't need that enzyme to synthesize our own folic acid. So it doesn't affect human much, it affects bacteria. So that's an example of competitive inhibitors. You can see, design something close to the substrate, and that will prevent the binding of the substrate to a bacteria enzyme to prevent bacterial growth. Another type of reversible inhibition is non-competitive. Non-competitive inhibition, like we said earlier, it does not bind to the active site. It binds to something else, somewhere else. For example, it binds here. Okay, that gray is a non-competitive inhibitor. When it binds there, the active site will change its conformation. The active site is kind of, hey, close. Then the substrate still cannot bind anymore. If that's the case, we call that non-competitive inhibition. It binds to a location other than the active site. So the enzyme structure is changed by the inhibitor. And the active site can no longer accommodate the substrate, so cannot catalyze the reaction. And this type of inhibition is actually very common in enzyme regulations in our biological system. It's one type of called allosteric regulation. We're going to see right away. Okay, say right away. So this is the first type of regulations of enzymes called allosteric regulations. What happens, you can see that enzyme has two sites. One is the active site, of course, for the substrate to bind and to catalyze the reaction. And some enzyme has another site called allosteric site. Okay, allosteric site. When a regulator binds to the allosteric site, the active site is going to change conformation so that substrate cannot enter. If that's the case, the enzyme is inhibited. See that? So this regulator, kind of like a non-competitive inhibitor, is called a negative regulator. It binds to the enzyme some place other than the active site. We call that allosteric site. It changed the conformation of the active site so that the substrate cannot bind. That means no reaction. So the enzyme what? Slows down. That's called negative allosteric control. Okay, very common, negative allosteric control. And sometimes you may even see positive control, means if the allosteric regulator binds to the allosteric site, the active site opens up for more reactions. So that means what? The allosteric regulator, in this case, the green one, promotes the reaction to make the reaction faster. And that's called a positive allosteric control. Okay, they both are very similar to the idea of non-competitive inhibition. This guy binds to some places called allosteric site, change the conformation of enzyme. If you close the active site, negative control. If you opens up the active site, positive control. Okay, positive control. Negative control, I think, is more common. Now, this regulation, okay, this regulation has a very, very important mechanism in metabolism called feedback control. Okay, feedback control. In feedback control, the final product will serve as the negative regulator for the first enzyme of the metabolic pathway. The final product will serve as the regulator for the first enzyme of the whole pathway. It means what? It means, for example, A to B to C to D, 3 steps, 3 enzymes. The D is the final product. Okay, final product. Let's say your biologics has a lot of D synthesized. Your biologics need to, hey, I need to stop. Don't make more D. I cannot use them anymore. Then D will do what? D will inhibit the first enzyme. What happened when the first enzyme is inhibited? The whole assembly line stops, right? Because the first enzyme stops, then they say you don't have much B, you don't have much C to go over to D now. So once you have a lot of D, D will tell the first enzyme, will regulate or binds to the first enzyme to stop the reaction, to negatively control the reaction. So the reaction slows down. But when D is slowed down, the D decreases. Hey, D is dropping. And hey, the cell says, we need more D. Then what does what? D will lose control of the first enzyme because you have less D now. Then D will kind of like get away from the first enzyme. Then the first enzyme will restart. The whole assembly will go on and give you more D. So it kind of like the D is giving what? Feedback to the first enzyme. Okay, first enzyme. So activation or inhibition of the first reaction sequence is controlled by the production product. Okay, this is product of the sequence. Okay, product of the sequence. And that is through here, through the allosteric regulation, right? Too much D. D will bind to the allosteric site to close the active site of the first enzyme. And then the reaction stops. When D decreases, this guy goes away. Okay, D goes away because D decreases. Then the site will open up and restart the process. Okay, that's a very important regulation of enzyme called feedback control. The final product will activate or inhibit the first enzyme in the sequence in the metabolism pathway. Another example of enzyme regulation. Okay, enzyme regulation is called zymogens. Okay, zymogens. Zymogens are basically inactive enzymes. Okay, inactive enzymes. They're synthesized, but they're inactive. They're kind of reserved in our cells. When needed, a small piece of peptide will be chopped off from zymogen to give you the active enzyme. And that active enzyme will be used. It will be used. A lot of proteolytic enzymes, I mean, enzymes catalyze the breaking of peptide bond, are zymogens. Okay, are actually stored as zymogens. Okay, a lot of protein, like digestive enzyme, protein broken, peptase are zymogens because they are synthesized, but we don't need them anytime. When you have digesting needs, you have more food taking in, then you need more of the enzyme. The enzyme will chop up a piece and ready to go. So they're kind of like a reserve form of the active form. When needed, this enzyme will, some other enzyme will chop off a small fragment to give you the active enzyme. Okay, so in a very fast way, instead of synthesized from the beginning. Okay, beginning. One very famous example of zymogen is shown here, and also related to drug development. Angiotensin is an octapeptide hormone. Octapeptide is 8-peptide hormone. It's a very short. This hormone is involved in increasing blood pressure by narrowing blood vessel, like vasoconstrictor. Okay, contract the vessel so the blood pressure increase this angiotensin. Angiotensin is a zymogen. Okay, is present as in the body as zymogens. They're actually stored in a 10-peptide form. Okay, in a 10-peptide form. When needed, 2 of the peptide will be chopped off by an enzyme called angiotensin converting enzyme, ACE. So when needed, this enzyme will chop up 2 peptides to give you angiotensin. To what? To increase pressure. Okay, to increase pressure. Okay, think about it. This increase blood pressure, BP. Okay, increase blood pressure. This enzyme will produce angiotensin to what? To increase the pressure. So think about it. If I want lower blood pressure, what do? We close the enzyme, right? If we don't produce angiotensin, then we don't increase the pressure. Then we what? We can decrease the pressure. So a class of anti-hypertension drug actually does this, inhibit ACE, angiotensin converting enzyme. Inhibit ACE means what? Less angiotensin. That means decrease pressure. Okay, ACE inhibitors decrease the formation of angiotensin. So what? Lower blood pressure. Remember, angiotensin increases blood pressure. But ACE inhibitor, that means shut down this process, less angiotensin, then lower blood pressure. And this classes of hypertension drugs is called ACE inhibitors or ACEI. Okay, ACEI. Okay, here I give you a list of ACEIs. Okay, you can see they all end with the same suffix pril. Okay, benazepril, captopril, mispril, and the parenthesis means their trade names. Okay, they're very commonly used not only to treat blood hypertension, but also treatment of cardiovascular and renal diseases, including heart failure, acute coronary syndrome, nephrotic syndrome, diabetes, and hypertension. So these are something involved in enzyme regulation sandwiches. Okay, so that's a shorter chapter. We talk about enzymes, which is a unique type of proteins, catalyzes all the chemical reactions in our biological systems. We talk about what are enzymes, how does enzyme works, the mechanism of enzymes, the active site of enzymes, the model of enzymes, and also the types of reactions it catalyzes, and the classification of enzymes. And also we talk about the factors affecting enzyme activities, and then we talk about enzyme inhibition and some cases of regulations. Okay, the chapter is a very huge one if you really study biochemistry, but I hope it will give you a very fundamental view of this unique type of protein, enzymes. Okay, that is all for today. Bye now.